You're not.
You're not?
Is he
You're not in
masked?
physics?
This was an open invitation.
Yes, very good. Yeah, so today I'm going to talk about really trying to understand what causes cancer, right? So that's what my lab is focused on. Our lab focuses on breast cancer, but it's like nice to take a step back and really try to figure out like what in the environment, what in what we eat might actually cause chronic disease. And like I said, like we said, when I was from the School of Public Health, this is our building. If you haven't been up to Ann Arbor, we have this very beautiful peony garden. It doesn't look like this right now, but should you come up to Ann Arbor, May or June, it's quite nice. Yeah, so some of you, if you're like sparkling water fanatics, like I am, so you might have seen this like hullabaloo in the news back in October. And so what like the gist of this was there's this lawsuit that was filed against the makers of the sparkling water brand LaCroix, right? It says like LaCroix contains the same insect found in cockroach insecticides, or the all-natural label on your LaCroix is meaningless. That doesn't mean seltzer is bad for you. And so it's like, what is going on here? Like, why was there all of this excitement over what we think would be innocuous, right? This like can of sparkling water. And this is language from this lawsuit, right? That was filed by this law firm, Beaumont Stales. And it says that testing reveals that LaCroix contains a number of artificial ingredients, and everyone's like, yes. LaCroix, in fact, contains ingredients that have been identified by the FDA as synthetic. These chemicals include limonene, which can cause kidney toxicity in tumors, linalool propionate, which is used to treat cancer, and linalool, which is used as a cockroach insecticide. And so you're like, you read this, and you're like, this stuff must be disgusting. It's like within a can of LaCroix, there's these cancer-causing chemicals. There's this chemical that's used as an insecticide. Like, what is going on? But if you like dig in a little bit deeper on this, it's like these are things that are found in food, right? Limonene. This is the chemical structure. Y'all are in a physics class. Maybe like taking some chemistry too on chemical structure. Limonene is a compound found in like citrus peels and citrus fruit, right? Like very aromatic. Linalool is commonly found in mint. And linalool is commonly found in things like cinnamon. So how this shook out is that this lawsuit alleged that LaCroix was using synthetic flavors. And of course, these are all chemicals, right? These are all chemicals with specific properties. LaCroix claims that these are from natural sources. But within this lawsuit is nested this idea that these, because they can be used in insecticides, and indeed limonene is used in a lot of different household products because it has that like bright lemony scent, right? It's used in a lot of like cleaning products, for instance. That doesn't mean it's like bad for us necessarily, right? It just is used for all these different things because it has these interesting chemical properties. And so this is what frustrates me a little bit as an environmental scientist, because we know that there are things in the environment that are going to cause disease, but we oftentimes, I think, focus on the wrong stuff, right? So probably like the trace amounts of these chemicals, which are naturally found in all the foods we normally eat, probably aren't the problem, right? And maybe some of y'all have heard of this guy, Paracelsus, who was one of the classic, sort of like one of the parents of the field of toxicology. And he came up with this concept of dose response, right? Where he says that all substances are poisons, there's none which is not a poison, and the right dose differentiates a poison from a remedy. And what that means is that no matter what the compound, if someone is given a high enough dose, sure, that can cause disease. Even something like water, right? Even something like water, we can drink too much of it and we can die from things like hyperhidrosis. So there's nothing that's safe, but it's really the dose that we're given defines whether something is toxic or not, right? And so this is a really important concept when we're trying to understand what in our environment causes chronic diseases like cancer. And so what we do in public health, in environmental sciences, is try to understand, okay, well, what are the most toxic chemicals that we might be exposed to on a daily basis? Let's figure out if we're exposed to levels of those compounds that might be hazardous. And so we use a process called risk assessment for this. And this is kind of like the very basic risk assessment framework that we as toxicologists do. So I'm a toxicologist by training. I teach our like intro-level toxicologists to the graduate students of U of M. And this is what we're teaching people. So first we want to see, like, is a chemical that someone might be exposed to hazardous? Right, we call this process hazard identification. So does the agent potentially cause some sort of adverse health effects, like my lab studies breast cancer, for instance. So could something cause breast cancer? Second, we want to assess, is there a relationship between the dose of a chemical and that adverse outcome? For instance, like what levels of this chemical exposure cause an adverse outcome? So that minuscule amount of limonene that's in your sparkling water, probably not going to cause cancer. But I'll tell you about some other stuff in our food that might in a little bit, right? The next thing we want to do is assess, what are people actually exposed to? Like, what are the levels of these chemicals that you and me, our kids, our grandparents, people in other countries are actually exposed to on a daily basis? And then we want to characterize the risk. Right, we want to see what's the incidence of these adverse effects? How often are people in our population getting breast cancer, for instance? How robust is our scientific evidence? How certain is our evaluation of this relationship between dose response and adverse effects? Are we adequately characterizing the most susceptible population? So people that might be more exposed, or people that might be more vulnerable to exposure, like those who are very old, those who are very young, especially pregnant women and babies. And then these are relevant mode of action. We like to understand the mechanism of what's going on. So as toxicologists, our job is to really characterize the dose-response relationship, right? And we do this with experiments in the lab. And usually what we'll do is we'll expose some sort of model system, sometimes cells, sometimes animals, to increasing levels of a chemical, and then we'll look at the outcome of interest. And so we'll build what we call this dose-response curve. Usually on the x-axis, we'll have an increasing dose. On the y-axis, we'll have some sort of response. And this is sort of like an old school toxicology graph, back when toxicology was a field where we would just give rats as much chemical as we could until they started dying off. It was like not a great experiment, but that's kind of what the field was like for 400 or 500 years. Not so much anymore. But we'll have like mortality on the y-axis, and then we can sort of like track across and see what sort of dose might be associated with our outcome of interest. We can then take that experimental data, right, that we identify from our rats, and we can try to estimate what might the risk in a human population be. And because we don't know everything, and despite what some of my toxicology colleagues like to think, rats are not just tiny humans, right? We have different biology, right? Mice and people are different, turns out. That's a hard sell for some people still. But we know that there's some species level differences, and so we can take account that species level difference by reducing that level exposure that causes an outcome. Maybe we'll divide that by 10, and then we know that people are different, of course, right? So there might be differences in genetic susceptibility, there might be differences in age, right? People respond differently to chemicals based on their age, and we can take that human variability into account. And so usually what it shakes out into being is that we'll identify a dose that causes the relevant health effect in an animal model, and then based on our understanding that there's differences between mice and humans and differences between all of us in how we respond to environmental stress, we'll divide that number by 100, okay? And so there's more sophisticated ways to do this, but this is sort of how things have been done in the field over the last 20 years. And like I said, toxicologists do a lot of different things as far as testing. So sort of the classic example is using animal studies, so mice or rats or dogs or sort of non-human primates. A lot of labs focus now on in vitro studies using cells, so that's a lot of what my lab does. I'll tell you a little about that in a second. And then there's a lot of toxicologists that now try to do everything on a computer. So can we estimate what's going to happen based on some fancy docking simulation studies, or looking at the structures of different chemicals and then looking how similar or different they are and estimating how they might interact with the biological system. And I think like a lot of other fields, there's a huge push in talks to try to reduce the number of animals that are being used in research. Let's push more towards in vitro and in silico research.
Question,
Yes,
is that for purposes of ethics or accuracy?
both. Yeah. So there's a lot of data that suggests that even between two rodent species, the concordance in response is only 60 to 70%. And so you would imagine that the concordance between a rodent and a human is not going to be any higher than
Right.
that, right? So there's issues with trying to estimate what's going to happen to a human based on what happens in an animal. Also, yeah, there's like, of course, the ethical component. Yes, I think that it's a lot. We're all like trying to figure out. There's also some interesting sort of like legislative pushes in the field. So Europe has banned animal testing for cosmetics, right? Which is great, right? But it kind of leaves us in a lurch because that's our like major way of assessing whether something is hazardous or not, has been animal studies historically. But a lot of like the legislation worldwide is starting to drive us away from animal testing too.
Thank you.
Yeah.
So if your lab focuses on one specific type of cancer,
Yeah.
but you also do general toxicology, do you have a rodent model for a rodent that would exhibit human breast cancer symptoms? Is that, do you have a
Yeah.
modified?
Yeah, so we have like a few different models that we like in the lab, depending on what process in breast cancer we're trying to study. So we have a genetically modified mouse that has a gene mutation that's similar to what women have when they develop HER2 positive breast cancer, for instance. And that mouse is much more likely to develop metastatic cancer. So that cancer like spreads throughout that mouse's body in a way that mimics what happens to a person when they're developing metastatic disease. So yeah, so there are some animal models that are still necessary because we have a really hard time modeling that process in vitro. So we can't really do that in a cell dish very effectively. This is something where we really need that like whole organism model still. Yeah, that's a good question. Yeah. Any other questions? So I started with the LaCroix thing because I think it's kind of silly and like there was like a big media splash and like I got interviewed about this and just like, this is ridiculous because we know that there is a lot of stuff in our food that's not good, right? And this is what really got me excited about the field of environmental health when I was in your shoes, right? I was working in a lab in Dallas when I was doing my Master of Public Health, so a little bit older than y'all probably, but my job was to go around to the different grocery stores in Dallas and buy food. And then we shipped that food off to our collaborator in Germany, who's a chemist, and he measured all sorts of different toxic chemicals in that food, things like pesticides and metals and flame retardants and plasticizers, and it was pretty striking. I'll show you some of these results, but let me take a step back and tell you about how we know, for instance, that metals can be extremely toxic. Maybe you've heard about some of these things. Of course, like the lead contamination in our neck of the woods up in Flint in the water supply. I'm sure everyone has heard about that. We know that Flint is a very potent developmental neurotoxicant. Cadmium is also something that's very often found in food, and we know that cadmium causes severe liver and kidney toxicity, and is also potentially a risk factor for lung cancer and maybe breast cancer. My lab's looking at this. But cadmium very much preferentially can bioaccumulate in rice. And so I work with a group out in Thailand called Mesat. It's like right on the border of Myanmar, so western Thailand. And this part of Thailand is located near a zinc mine. And so the zinc mine had very poor industrial controls. And that zinc mine sort of like runoff flooded into this creek, and then all around this are rice paddies. And so if you've ever seen a rice paddy or seen pictures of a rice paddy, you know there's like a lot of water, right? So all of that mine runoff water flooded these paddies, but that mine runoff water had a ton of cadmium in it, unfortunately. And so that rice preferentially bioaccumulates that cadmium. A lot of the people who live in this part of Thailand are subsistence farmers, right? So they're eating what they grow. This is how they're surviving. So they ended up really highly exposed to cadmium from their rice supply. This also happened in a very famous case in the Toyama Prefecture in Japan in the 50s and 60s. And this is a picture of a poor individual who was highly exposed to cadmium at that time. And it caused like this like severe bone toxicity. You can see how they're like hunched over, right? And the doctors in Japan at the time named this disease a tai disease. A tai translates into ouch ouch disease, because these people were in like so much pain, right? And so this ends up being a huge issue. So this is like one way that we know that there can be toxic chemicals in our food. Another classic way that's more relevant to what we might be exposed to today are these perfluoroalkyl compounds, or PFASs or PFAS. Has anyone heard about this? This has been in the news like a lot recently. I've seen some nods. So these are chemicals that have really interesting properties. They're used as surfactants, which means that things like don't stick to them. So things like Teflon, right? You can like fry an egg in a Teflon pan, it just like comes right out. Or like Gore-Tex on your jacket, right? Where you have like these waterproof coatings where like water just slips right off. It turns out that these chemicals are extremely environmentally persistent, and they can cause severe toxicity. If we look in people's blood, like we could take any of our blood from anyone in this room, and we'd be able to detect these chemicals in it. Right? These are all over the place. They have a really long half-life, so chemical half-life of these is quite long, and their exposure is associated with all sorts of hazardous outcomes, like kidney cancer, thyroid disease, reproductive outcomes. Even in Ann Arbor, right? So we have the Huron River goes through Ann Arbor. We have a fish advisory. We're not allowed to eat the fish in Ann Arbor because those fish are contaminated with these chemicals. So this is not a situation that's unique to Michigan either by any stretch of the imagination. This is like the big and emerging environmental health concern, I would say.
So question.
Yes.
So is that okay if we use a pan coated with this type of?
So I would recommend not, yeah, because that's like a way that will transfer directly to your food. So like stainless steel pan, I'm like personally a cast iron pan fan where you can still get like sort of a natural nonstick. Yeah, especially, sorry.
Oh, no, I was going to say exposure on cooking with that. I'm just curious. Do you want to tell them about what the exposure is based on there's some of the studies they've looked at? I didn't know if you want to talk about that.
Yeah, well, I was gonna go on, but yeah, so.
No, sorry about that.
No,
Dan,
it's fine, no, it's fine. So, yeah, so what we know is that especially high-fat foods, right? Like this sad onion, if you're cooking it with oil, some of the PFASs can transfer through there. But really, what you definitely should not do is like chip off that coating. It's like, don't use those like metal utensils on the non-stick pans. That coating will come off in your food, and then you'll just be like directly ingesting all of these chemicals. And remember, these chemicals are why we can't eat the fish in the Huron River in Ann Arbor. So you definitely don't want to be eating them in your house, right? Did you have anything?
Thank you. No, thank you so much. That was what I was hoping you did.
Yeah.
It increases exposure.
It increases exposure dramatically. Yeah. Yeah, so those are PFCs. I'll provide these slides for you guys if you're interested. So the Environmental Working Group does this great survey of pesticide residues. They kind of collate data that's generated by the USDA. USDA will do one of these like market basket studies. Well, they go to the grocery store, they'll buy different fruits and vegetables, and they'll look for different pesticides. And then the Environmental Working Group goes and they identify so like which fruits and vegetables tend to have the highest levels of pesticides. They call them the dirty 12. So, you know, of course, some of the best fruit, like strawberries and nectarines and peaches. But then you have like the clean 15. But you can go to this website and sort of like see what's in what you eat. Right. This is really important. Question?
Do you happen to know how much of a difference it actually makes paying more for organic, especially like with the dirty 12? Do you get organic strawberries? Are they actually going to test lower in some of these?
They might have different. So there's even organic pesticides right, they'll have a different set. They'll be lower. Nutritional content doesn't seem to be super duper different. But yeah, so you can avoid some of these by eating organic. Yeah, you might be exposing yourself to some things that we don't know that help that stuff as much. Yeah, but you won't be exposing yourself to the things that we know are super toxic. Yeah. And then another classic one that folks in Michigan have been interested in a long time is BPA, bisphenol A. Anyone heard of BPA? It's like in your water bottle, they used to be like the water bottles, right? People freaked out because it's what we call an endocrine disrupting compound. So that means that it looks enough like a hormone in your body that it's going to activate your endocrine systems. And so my lab's really interested in the case of breast cancer because it looks enough like estrogen that it can trick breast cells into growth. Right? And so BPA was historically in sort of like Nalgene bottles. They've moved away. Now that BPA has been switched out with some other bisphenols, like bisphenol F or bisphenol S or bisphenol Z or bisphenol A, F. So like all very like similar compounds with actually similar effects. So again, it's like metal water bottles. Go. But this graph on the right just looks at how BPA exposure influences mammary gland development in a rat. Right. So it really like promotes a process like early stage breast cancer when mice are exposed to BPA in utero. So it's like these like early life exposures to BPA seem to really predispose a rat to getting breast cancer later in life. So we know that this is in a lot of food, of course, the lining of tin cans. And phthalates are another important compound that end up in food very often. These are used to make plastics sort of like flexible, right? And so you can think of this like these like gloves or sort of this like conveyor belt are made of phthalates or this like tubing. And these also have effects on your hormone system. So they look enough like testosterone that for the guys, they might inhibit how androgen receptor works. Also, developmental toxicants seem to be pretty strongly associated with preterm birth and placenta dysfunction. And so we know that these can be detected. This is some work that we did. These can be detected in food you buy from the grocery store too, right? And we also know this is an interesting study from Amy Zoda's lab at George Washington, where she looked at the relationship between phthalates and fast food intake. And what they found was that individuals who report eating more fast food have more of these phthalates in their body. Right, so it's like something about how those fast foods are being processed leads to more of those chemicals getting into their body and potentially causing those adverse health effects related to phthalate exposure. So some of you might be aware that if you've like been to California, you know like everything is labeled as a carcinogen or not based on Prop 65. So some of these phthalates are known carcinogens based on California's definition. And then another classic example that we don't run into that much in America, but we see a ton in Mexico and we see a ton in Asia, is using lead glazed pottery, right? And this ends up being a huge route of exposure to lead in countries where storing food in lead glazed pottery is common. So I have some colleagues that run a study in Mexico who are really interested in early life exposure to lead. And what they found was that whether you store your food in these lead glazed pottery containers or not is one of the best predictors of an individual's lead exposure. So even just like think about like what are people keeping their food in, especially if that food is acidic, it's like leaching the lead out of that glaze. And that lead is getting into the food, and then that's getting into, in the study that my colleagues are interested in getting into the moms and then getting into the babies while they're developing. Of course, we know that like lead can be a very potent neurotoxicant, right? And so we've done a lot of work. I was telling you about how when I was doing my master's, I went around to the grocery store and I bought a bunch of different food and we measured all these chemicals in the food. And we had like one wild finding where we had done an initial experiment. We didn't have a ton of money for this study, so we kind of like pooled different types of food together and then averaged like measured like chemical concentrations in that like food pool. And we got concentrations of brominated flame retardants, so the things that are in like these chairs that help them not catch on fire, which are also look like thyroid hormone. We found that those seem to be really high in these butter samples for some reason. For some reason, these butter samples had just like tons of brominated flame retardants in them. And so we like looked at our data when it came back, and we asked the chemist to like go back and analyze the individual butter samples to see if what was going on. And so our chemist colleague went back and analyzed 10 different butter samples and found that one of those butter samples was 1% flame retardants. That was like something I bought at the grocery store in Dallas. It was 1% flame retardants, like the butter. Like, and so that like obviously made some news, right? So we like, CNN picked us up, Scientific American picked us up. It's just like, oh my God.
Did it taste any better?
I don't know. I mean, it might help you, like, maybe you become flame retardant. I don't know, like, we reported it back to the company and they were not so receptive. I don't know. But this is just like the stuff that we like focus on interesting things, I guess, is what I'm trying to say. People are like all freaked out about whether there's these like, what we're adding is flavor additives like limonene, like that's in a lemon. Like everyone's concerned about whether that's bad. When we find that we can detect a stick of butter that's 1% flame retardants in the grocery store, it's strange what people worry about.
What
Yeah,
about
this
that
is just
peanut
like-
butter? Did you guys look at any of the aflatoxins in that or not?
No,
Is
we
that
did
out?
not do any
Okay,
aflatoxins
good.
on this
I was
one.
just
No,
curious.
that's the
It just
great-
adds to the butter fun, yeah.
That would have been an interesting
I
add-on.
know.
Yeah, and so this is like this is a paper we published, looking at these flame retardants, these like different types of flame retardants, and what we find is that we can see these, they're mostly in dairy, they're mostly in meat, and so that makes sense to us because these compounds are fat soluble, right? They're what we call lipophilic. When they're in their body, when they're in your body, they bank in the fat. They also are attracted to fat. So if you eat like a lot of high fat foods, you might be more likely to get these sorts of chemicals in your body. We did a similar study with some like old school pesticides, which was like terrifying to me. So has anyone ever heard of DDT? Yeah, you're nodding. So this is like an organochlorine pesticide that was used pretty extensively in the 50s and 60s, right? By the 70s, we started to know it was really toxic, right? It was causing some big time effects in, especially like predatory birds. It was causing thinning of the eggshell. Maybe some of you all have read Silent Spring by Rachel Carson, if you're more environmentally minded. All about these types of compounds. Almost all of the food that we purchased at the grocery store in Dallas, and this was like 10 years ago now, but you know, 2010-ish, all of it still had DDT in it. So that stuff persists through and is still detectable. Again, it's like something we could measure in all of our blood, and we would see DDT in all of our blood stuff. And so, a concept that a lot of the folks in my field are starting to come around to is this idea of the exposome, where we're starting to realize that we are exposed to a lot of different chemicals that could have adverse health effects, but at low levels, and we don't really know what we should worry about still. And so this was a nice article on the cover of New Scientist. It says, we've made 150,000 new chemicals. We touch them, we wear them, we eat them. Which ones should we worry about? And so this is what this concept of the exposome tries to understand. It's like, how do we like comprehensively assess this totality of exposure that all of us are getting on a daily basis, whether it's through the stuff that we eat, or whether it's through the personal care products we put on our skin, or like the air pollution that we breathe, or if you're up in Ann Arbor, the PFASs that are in the water that we're getting from the Huron River, right? So how do we understand the health effects of all of these different types of chemicals? Why I'm really interested in this with the context of cancer is that we think that cancer is only about 5 to 10% genetic. We think that the other 95% of cancer risk comes from some sort of environmental exposure. And so this was an older review article, but I really like this figure because we can break out that environmental component, and we see that there's large impact of diet, of course. So we all know that diet is a really strong risk factor for cancer. Of course, tobacco smoking, we know that that's bad, right? Infectious diseases cause cancer. Does anyone know an infectious disease-related cancer? Cervical, yeah. So that's why everyone gets HPV vaccinations now, right? Obesity is linked to cancer. Probably now we know that this obesity piece of the pie is bigger. Alcohol and others is kind of like what we're interested in. Like, what is this like chemical mixture that we're all growing up in doing to our bodies, right? And how does that influence cancer risk? And so I want to just tell you just like a smidgen of what my lab's up to in this space, just to like wrap this up, where we're trying to figure out, you know, what are people exposed to, and then are these chemicals hazardous, right? And my lab's major focus on our big grant that like keeps the lab funded is on the area of cancer disparities and trying to understand why African American women are so much more likely to get triple negative breast cancer, the super aggressive subtype of breast cancer. So what we really want to do is try to understand what are chemical exposures in very susceptible populations. And so to do this, we leverage data from an ongoing study that's run by the CDC. It's called NHANES. It's the National Health and Nutrition Examination Survey. So this study initially began in 1959. It's been running in some iteration ever since then. And what they do is they go around to different counties in the US and they recruit around 10,000 people a year. And they bring those folks in and they bring them into these like CDC trailers that they set up that have all of these different rooms where they're doing this like really comprehensive workup on all the study participants that come through. And really what they're trying to do is determine different disease prevalences, determine the nutritional status, and measure a bunch of environmental risk factors for disease, trying to get like an idea of what's going on in the US population. What's cool about these data is they're all publicly available. So any of you, if you're interested in doing this, you can go download all of these data. These are available for anyone to use, which is great. Right, so the CDC makes all of these data available. What's great for us in the environmental health field is that they measure chemical biomarker levels of about 400 different chemicals. So these are like metals, pesticides, air pollution, volatile organic compounds, those plasticizers, things like this. And they have like measurements of all of these different chemicals in people from the US. And it's a nationally representative sample, which is neat. So we can like figure out like who is exposed to what and at what levels, which is really important. And so this is a paper that one of my very smart PhD students, I'm like blessed with smart PhD students, but BUN, she's a bioinformatics PhD student at U of M, and she downloaded all of this exposure data. So this is like 400 different chemicals measured on like 80,000 people. So it's like a pretty big data set, right? Good chunk of data. And you can see kind of like what's in this data set. It's a lot of things like volatile organic compounds and metals, some of these like old school chemicals like PCBs. We're able to like estimate the half-life of these different types of chemicals in an individual's body. So some of these chemicals like PCBs have a half-life on the order of 5 to 10 years. Some of the chemicals that a lot of us tend to be exposed to these days, like those plasticizer phthalates, have very short half-lives, like less than a day. Right, so if we're detecting those in a person, that means we are constantly exposed, right? Because our body excretes those very quickly. We can also look at the relationship between age, an individual's age, and the levels of chemicals in their body. And so as you fall out more to the right on this plot, that means those chemicals are found in older individuals. And if you fall out to the left on this plot, it means they're found in younger. So individuals that were older tended to have more of these like organochlorine pesticides like that DDT, these like PCBs, things that have been phased out or made illegal because we know they're so toxic. They still are found in pretty high levels in the bodies of older people that were alive when those exposures were high. And it's just like the half-life of those chemicals is so high you just don't ever get rid of them. So like they're still detectable in their bodies. For the rest of us, and especially you guys, these other chemicals are gonna be of more interest, right? So things like parabens, which maybe you've heard of in cosmetics, maybe they'll use it as preservatives, maybe it's like paraben-free cosmetics at the grocery store. Phthalates, these are like the relevant exposures for younger folks. And so V came up with this funky idea of how to like understand what are chemicals that old folks are exposed to and what are chemicals that are young folks that are exposed to. And she made this like beautiful plot where each of these individual dots is a chemical. And as we like move to the right on this plot, it's things that older folks are exposed to. And just to give you an idea of what this looks like, we can look at this type of PCB, and we can see that here's like levels in young people versus old people. And you can see like, oh, okay, there's like a huge difference, right? This is like a chemical that you only would find in old people. But conversely, there's chemicals that are only found, chemicals that are found at much higher levels in younger individuals. And so one of those is like one of those phthalates. So these are used as fragrance carriers often in cosmetics, and we see that teenagers and kids actually have the highest levels of these chemicals. Right? So it's like interesting trying to understand, like, based on what we consider these demographic characteristics like age, that like chemical exposome varies. Right? So the chemicals that are in someone who's older's body look very different than the chemicals who are in someone who's younger. And so this is just a cute figure, but she like was able to draw this line and be like, okay, well now we can like characterize what might be in an older individual versus a younger individual. If you're interested in this paper, just shoot me an email and like we have to send you a copy. And then sort of like the last part here is how do we try to understand the impact of all of these different chemicals on an individual's risk of developing a disease like cancer? So we know that cancer, again, is like 90 to 95% environmental. Do any of these chemicals play an important role? And so, like I said, my lab is really interested in understanding breast cancer disparities. That's what our big grant is on. And there's this aggressive type of breast cancer, triple negative breast cancer. There's no treatment for it, where women of African ancestry in the US are 2 to 3 times more likely to get it, right? And we don't have a great idea why still. And so V and a very bright undergraduate in my lab, they went back to that NHANES data. They're like, well, are there differences in the chemical exposures in the US on average in women who are non-Hispanic Black versus non-Hispanic Black? Got a question?
Have you compared that to women living in Africa?
Yes.
Are they 2 to 3, are American, are women of African descent in America 2 to 3 times as likely as their counterparts in Africa to develop that cancer?
Yeah, so that's a great question. So University of Michigan has this ongoing collaboration with scientists in Ghana. So Ghana is like Western Africa, right? And so we find that triple negative breast cancer rates in non-Hispanic white women in the US are about 10 to 15%. Non-Hispanic Black women are somewhere like 20 to 25%. And then women in Ghana, at least at the hospital where our collaborators work, were around 80%. So it's like, there is this like gradient, right? And so we're trying to understand, could there potentially be an environmental component of these cancer disparities? And so I asked V, bioinformatics wizard that she is, to like crunch all that data and figure out what chemicals are different, like the most different between non-Hispanic Black and non-Hispanic white women in the US. And so you can see this is like this is like a pretty good chunk of data, right? We had data on, like 7,300 non-Hispanic Black women, about 9,500 non-Hispanic white women. And then these are, this is called a volcano plot, if you've never seen one of these before, because it kind of like explodes up like a volcano. On the x-axis, things that come out to the right are higher in non-Hispanic Black women. Things that come out to the left are higher in non-Hispanic white women. Like the further away it is, like the bigger the difference. And then the y-axis is a measure of statistical significance. So like the more statistically significant, the further up it is on the plot. And you can see that, so like for instance, benzophenone 3 on average is like on average 2 times higher in non-Hispanic white women. That's like the active ingredient in sunscreen. It's a UV blocker. That like makes sense, right? But then we can look at like what might be higher in non-Hispanic Black women. It turns out that it's things like methylparaben, propylparaben, a lot of stuff that's found in personal care products and consumer products, things that cosmetics, things that we might be putting on our skin, right? And so these are different in a relatively substantial way, 200 to 300% higher on average. So you're like, there could potentially be differences in the chemical, so like this chemical exposome that might be driving these cancer disparities. And so like, how do we figure this out? So like, how can we assess this? And so this is what my lab's up to. So we're trying to grow up what we call a biobank of normal breast tissue from a diverse donor pool. So women who are of African ancestry, women who are European ancestry, women who are of Asian ancestry. Let's get as many of these samples growing up in the lab as we can as like cell lines, right? We can grow them up in 2D here. To do this, we're partnering with Susan G. Komen, who runs this amazing normal tissue bank. And so this is a study that's run through Susan G. Komen, where they recruit women in to give voluntary breast punch biopsies. Eight voluntary breast punch biopsies. These are healthy women. This is me and my graduate student Shanice working one of these tissue collections in Detroit. We were at the Carmanos Cancer Institute, where we had 180 women coming in afternoon to give breast punch biopsies. And so some of those get frozen down for like RNA, protein, DNA analysis. Some of them get put in paraffin so we can look at them under the microscope. Some of them get cryopreserved so they're still alive, so then I can get them back in my lab and we can grow up like normal cell like that. Like keep them growing in a tissue culture dish to do experiments with. Question?
So kind of back to that, I read something recently that a huge number of our cell lines that actually have misidentified because they were using people's backgrounds. Send them to do genetic analysis that was completely different.
Yeah,
So
it's
are you
a
guys
huge problem.
characterizing this genetically or based on real time?
Yeah, so I don't have any, has anyone ever done a tissue culture experiment before? Has anyone ever worked with a cell line here? Yeah.
Yeah.
So like the classic version of this story is the HeLa cells. It's like the first cell line, right? This like very aggressive cervical cancer cell line from a woman, Henrietta Lacks. And that line like contaminated a ton of tissue culture lines like in the 70s and 80s. This is still a persistent problem, right? There's a huge issue with misidentification. So for these, we're like genotyping everybody with like a SNP array, and then we're also doing STR analyses. But for those of you that end up doing tissue culture, this is like not something to be ignored. This is like an extremely important issue. Even within breast cancer, there was like one very popular breast cancer cell line that people have been using for years and years and years and years and years. Turns out it was a melanoma cell line. Oh, cool. Like, no, I didn't do it. Yeah, so this like characterization issue is extremely important. Yeah, so we're in the process of like growing up this biobank of normal breast tissues, so we can like start to do experiments on them. And like what we like to do and what Evan Hill in my lab is working on. Evan's mom. Is like, can we model mammary gland development in a tissue culture dish, right? And so we can grow these guys in these 3D conditions that mimic the extracellular matrix of the breast, and we can mimic breast development in a dish, which is super cool. Oh, my video stopped. Let's see if I can get it back again. Oh, stop playing. That's fine. It's cool, it's fluorescent and gross. I don't know. But yeah, so we can like model like the actual like development of a mammary gland in a tissue culture dish, and then we can like do this in the presence of or absence of different chemicals, especially those ones that we prioritize based on exposure disparities. Like, see, does it really impact pathways that we might think might be associated with breast cancer, right? So we can see like basic stuff like, does it increase how cells grow, like their growth rates? Does it change protein expression in the cell that we might think could be associated with cancer, or maybe some other like growth characteristics? Does it change gene expression? We're really excited about like single cell RNA sequencing, super fun. But ultimately, can we like do these like dose response assessments to figure out what are the relevant doses of these different compounds, and eventually the relevant doses of these mixtures of compounds that are going to cause disease, right? That might be associated with breast cancer risk. And can we explain the biological basis of cancer disparities? Is the goal. Yeah. So in summary, we're exposed to a lot of stuff through a lot of different ways. Sparkling water is probably not the boogeyman here. There's a lot of things to worry about, not that one. Chemicals have multiple uses. You can use those as a flavoring agent. It might also be an insecticide. At different, at different doses, it does different stuff. And toxicity is a function of dose. And what toxicologists like me do is to conduct experiments to try to like assess which of these chemicals are going to be hazardous and at what doses they act, and kind of understand like what's the mechanism by which these things act. And so this is my lab, or most of my lab, at the recent Michigan SOT meeting. They do like all the work. I don't do anything. I just like grants, talk, yes. Teach classes. And so I just want to like highlight our program real quick. If you think that like this environmental health stuff is cool, I'd recommend you check us out over at U of M. I don't know what this is supposed to be. Okay. Weird. Yeah, so our department really spans the gamut between trying to understand human chemical exposure to doing like the molecular hardcore toxicology like my lab does, with like also a focus on industrial hygiene and worker safety. So like we like sort of like span this gamut about really understanding what's happening with the environment and human health. We have a couple different degree programs. We have a Master of Public Health, sort of like a professional degree program. We have a Master of Science, so a research degree program. We have a PhD. So like the students that in my lab, I did my PhD in this department. It was fun. I can vouch for it. I think with that, I'll take any questions you all might have. Thank you so much again for the invitation to come talk. This has been
Great
really fun.
presentation. Super job.